Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
Okay, good morning. It's a pleasure to have Ben Hall with us today. Ben did his PhD in Oxford with Mark Sampson, where he worked on development of new computational tools for new computational methods to study dynamics and function of large molecular structures. And then he continued to a postdoc at the Center for Integrative Systems Biology, directed by Judy Armitage, where he worked with Judy and others on the studying of bacterial chemotaxis, signaling. And I think this is also the topic of his talk today, so I'll let you get on with your talk. Thank you.
Thank you very much. Good morning. So today I'm going to be talking about some of the work I carried out in Oxford relating to multiscale modeling of bacterial chemosensing. And specifically today, I'm going to be talking about modeling the model bacterial chemoreceptors, TAR and TSR, with two different types of approach. One is a high-throughput strategy to understand large bodies of mutagenesis data that have accrued over the past 30 years in the literature of just the transmembrane regions of these proteins. And alongside this, very much larger structures, simulations of structures, of complete structures of the entire system. I'm going to talk about what exactly those relate to. And the key thing I've been trying to do here is to relate the dynamic behavior of both the small components in the complete system to the function and how the signals are passed along the length of the protein. I'm going to start off, however, by giving a little bit of biological background on the system I'm interested in. And the system I'm interested in is bacterial chemotaxis. Now, this is the response of a bacteria such as coli to its environment. That is, the technique by which it moves towards food sources and away from toxins. And this is achieved by a biased random walk, by alternating between running behaviors where it swims in a single direction and tumbling behaviors where it randomly rotates into new directions. It's able to bias its motions towards the food sources and away from toxins. And this is achieved in the cell through a pathway indicated here for E. coli, whereby a binding event at the receptor domain of the chemoreceptor is communicated across the cell membrane into the cell and causes either activation of the kinase Ki-A, if it's a repellent binding, which then causes downstream phosphorylation events and ultimately cell running, or in the case of attractant binding, causes the inhibition of the kinase Ki-A, which then leads to leads to cell running. So it's alternating between these running and tumbling behaviors, which allows the cell to go to one source rather than the other.
So the tumbling, is that, does the cell do something to tumble?
Yes. So what happens is that, so in coli there are about 3 or 4 flagella per cell, and what happens is by rotating in one direction, they determine if it's going to run, because the flagella filaments, which are long rod-like structures, arrange into a single body and then propel it in one direction. When they rotate in the opposite direction, they cause the cell to tumble because this bundle, which is coalesced, comes apart. They all fray off into different directions, and the cell changes its direction. And that's achieved by downstream activators such as Ki-Y phosphate, which is phosphorylated by the kinase Ki-A, which has been in turn activated by the chemotaxis proteins. So there's a complex system even in the model cell E. coli. TSR and TAR are the serine and aspartate chemoreceptors, and they're really models for the whole bacterial the methyl-accepting chemoreceptors, but also bacterial signaling in the general. So they really have been, they've been studied extensively over the past 30, 40 years, but also they can be used to give insights into other systems such as bacterial antibiotic resistance and other things. And in this specific case, I've been looking to model mostly the structures just from E. coli, because for this particular system, there are most of the structures and most of the components available.
In your previous lecture, the chemoreceptors are the bits on the surface?
Yes, the chemoreceptors are just the bits on the surface.
That's the bit you're focusing on?
That's the bit I'm focusing on, that particular aspect. And the key thing we were interested in within these chemoreceptors is how a signaling event goes from the receptor domain here through a very small motion of about 0.15 nanometers along the length of a protein through the transmembrane region, through a HAMP signal transduction domain, and ultimately to a coil-coil domain, which then binds and affects the downstream effectors over 25 nanometers. So there's a small motion is communicated, a small motion of about 0.15 nanometers is communicated over a 25 nanometer protein. We really want to understand this signaling event. And what we have in terms of data is we have structures of the receptor domain with the ligand bound and unbound. We have some low-resolution disulfide mapping information on the transmembrane domain, which is less useful, but it's still data. We have a NMR structure from a related HAMP domain, and we have a structure of the coil-coil domain. And what we know through mutagenesis and also through cell activities within the cell, all of these domains can be altered to change the signaling through the system. So we can turn the cell off by mutating any one of these different components.
When you're talking about structure here, you're talking about physical.
I'm talking about physical atomistic structure.
Exactly how the atoms are laid out with respect to each other.
Exactly, yes.
Sort of chemistry, as it were, I suppose.
No.
It's part physical.
This is the physical. So I'll talk about the models in a minute, but yes, everything is represented explicitly, and we're looking at the particular atom positions. And I think really the key of what we're trying to do here is to connect the various disparate data sources that have been generated over the past 30 years in a predictive fashion. And so these go from the schematic level diagrams where we've got information on the domain positions and the domain orientations. We have high resolution information on the physical structures of the individual domains disconnected from the complete chemoreceptor. And then we have low resolution information on the complete system. And really what we're going to be able to do is to take this sequence level data, this high resolution data and this low resolution data, and come up with a model which is both coherent with all the data that's available, but also is able to predict new things that can be gone and tested by experimentalists or can be tested by further simulation. And really the two approaches I've got for this involve trying to address this aspect of how the different scales relate to one another. So one approach has been to iteratively build bigger and bigger models of the system. From the individual structural components which you have available, trying to build models of initially the complete length dimer, which is represented here. In the middle, from the individual pairs of domains and multiple domains by exploiting overlap, and building bigger and bigger systems and seeing how they interact with one another and how they behave on their own.
What's a model in this context?
In this context, I'm talking about a structural model, so a proposed structure, physical structure of the chemoreceptor.
3D space.
Yes, that's correct.
So in one of these coils, how many atoms are you talking about? Millions?
It depends on the representation. It depends on the representation. So we're generally going to be talking about coarse-grained simulations, and so we're talking about thousands and tens of thousands of particles. For the biggest systems we're looking at, we have 50 million particles, and we have to include all the particles because it's a multi-body problem, and so we explicitly...
Okay, so a model has 50 million particles located in 3D space.
Yes, that's correct. And alongside this, we've been trying to address lots of data there is on just one component of this, that is the transmembrane region, by just simulating just individual components in a realistic environment, and then in a high throughput regime, which allows us to get very good statistics on the behavior and understand in detail exactly what precise motions are being undergone. And the hope is that we're able to draw these two different types of approaches together to get an integrated view of how this structure works as a system, how the individual components relate to one another, and how signals are passed through from this receptor domain to the coil-coil region. The key tool we've been using here is molecular dynamics, and in particular we're using what we know as coarse-grained force fields. These refer to the Martini and Bond force fields, which have become increasingly popular in MD in the past few years, and reduce the system size from about 4 non-hydrogen atoms, that's carbon, oxygen, sulfur, nitrogen, to one coarse-grained particle. This increases the speed of the simulations massively because it means there's a quarter of the number of particles in the system there would be compared to a detailed system. But also it smoothens the energy landscape. Because there are fewer particles, the energy is more quick to, it's quicker for the structure to explore conformational space. And the really objective here is to, just to reiterate, is to generate predictive insights into the macromolecular behavior. So we know that the complex macromolecular structures can link structure function through the dynamics, through the behavior over a period of time, and this changes in structure over a period of time. And what we want to use is the dynamics of the models on distinct scales to understand the behavior of the structure as a whole, structure as a system, really. So I'm going to start off by talking about the high-throughput simulation of just transmembrane components. In order to perform this kind of simulation, we've developed a novel software specifically for performing high-throughput simulation. So what we've developed is a tool I've called Sidekick, which is a software package which sets up massive numbers of simulations based on a limited set of information. So that is, it creates models of the structure in a realistic environment from an input sequence or a set of input sequences or an input sequence and a scanning mutant, as would have been performed in the experiments. And then all of these simulations are run over a high-performance computing grid. And the real strength of this is that it automates everything. That is, from an input sequence, it will generate the system, it will then run the simulation in a robust fashion, checking for crashes, and then it will analyze it for you.
What's a simulation? Yeah?
A simulation is an...
Like a model, which is one of these 3D things.
Yes. And this,
You jiggle this or something?
yes, so it's in molecular dynamics, so we're iteratively solving F equals MA to generate new positions after a period of time. We have to integrate numerically because it's a multi-body problem. So what happens is we generate a trajectory where we see a series of the structure changes over a period of time in terms of the model changes its behavior, but also water is able to move around it and lipids are able to move around.
So if you kick the strand at one end, then you follow it?
Yes, or it may follow through. If you perturb one end, you might expect it to, and that would be one of the things you look to test. That's what individual simulations do consist of, yes.
So you run multiple simulations because you're trying to perturb it in multiple ways?
Yes, but also we want to get very good sampling. So one of the issues with, especially with the large systems I was talking about with up to tens of millions of atoms, is you can only run them once because they require massive supercomputing resources to run them. Whereas with these small systems and an automated pipeline, we're able to run this massively in duplicates. So we can both search sequence space for lots of different mutants, but we can also do multiple replicates to ensure we're getting a realistic behavior. Okay, so to break down the pipeline itself, what we start off with an input sequence. From that, we're able to generate a model of the structure of the structure of the helix in a coarse-grained force field. We then place this helix in a box containing water, lipids, and counterions. And initially, this water and lipid and counterions is all mixed up randomly. So despite the fact that we're interested in a bilayer, we don't start off with the bilayer. We allow the bilayer to naturally self-assemble over the period of the simulation. And sorry, pardon? The bilayer. So the lipid. So the cell is surrounded by a lipid bilayer, and that lipid bilayer has specific properties, including a hydrophobic core, an interfacial region which both has elements of hydrophobicity and hydrophilicity, and then there's outside the membrane itself, which is water. And the transmembrane region I'm interested in simulating is specifically within that bilayer. The bilayer consists of two layers of phospholipids. So the phospholipids have hydrophobic tails, which point into hydrophobic core, and hydrophilic polar head groups, which point onto towards the water. And these bilayers really maintain a separation between the inside of the cell and the outside, and so any signal has to pass through the bilayer.
So you recompute the bilayer every time?
Yep. Every simulation effectively has a different bilayer. And the advantage of this is then it reduces bias, because if you self-assemble a bilayer, there's no way of predicting you'll get an even number of, for example, lipids on either side of the bilayer, or you get the same bilayer every self-assembly. And so we've done this self-assembly to reduce the bias in the system. So we're putting less, apart from the initial position of the helix, we've got the bilayer is not the same, and so there should be a greater degree of variation. So then, based on, with this system, then we then perform what I've termed robust MD simulation, which basically means that we run the simulation through standard MD packages such as Gromax. We then check to see if the simulation has crashed. And if it has crashed, then we restart the simulation from the last good time point with slightly more gentle parameters. And then we continue the simulation from that point, finally concatenating all the results into one continuous time trajectory at the end of it.
Why would it crash?
So the thing about the feature we have with the coarse-grained force fields is we can't always predict the appropriate time step for MD. So MD requires several inputs, one of the most crucial of which I think is the time step. So what you want to have is a time step between individual iterations of the integration of the Newton's second law. And you need to have a time step which is shorter than the fastest motion of the system. With the coarse-grained force fields, because they're relatively new, we're not always sure what the right time step will be for a given system and for a given simulation. And so we can't always predict from the offset which we would want for a particular system. So what we do is, when I say more gentle parameters, the key thing we do is we reduce the time step to about half. Now the downside to that is it means it runs half as fast, but if it does run. Whereas if we have a faster time step, we're able to explore more conformational space more quickly. Based on this simulation of just the helix in a bilayer, then we analyze it based on key metrics describing how the helix sits in the bilayer in terms of its position, orientation, and likelihood to appear in the bilayer in different simulations. And then we can then mutate and replicate. That is, we can repeat this cycle for different helices or for different random input seeds. So we can effectively replicate this and get very good statistics for each of the behaviors. And the strength of this is, whereas previously people would be able to maybe run a single simulation of your protein of interest, see how it behaved, as time has gone on, people will then try to expand this to looking at multiple ones and comparing these behaviors back to biological activity. And then furthermore, people will, if they had the time and they're particularly enthusiastic, they may have tried to repeat these to get good statistics. The problem with this kind of approach, however, though, is it's very time-consuming. Management and curation of the data as well as analysis become a problem when you've got large numbers of disparate simulations which you all have to analyze individually. With the Sidekick automation, what we're able to do is this on at least an order of magnitude or 2 orders of magnitude larger scale, and we can also automate this all through a web interface. So we can run massive numbers of simulations, generate massive amounts of data just from typing in the sequence, telling the sequences, and pressing go, essentially. We're bringing it back to the bacterial chemotaxis system. The particular helix we're interested in connects the receptor domain to the signal transduction domain, the HAMP domain, so crosses the bilayer. And this is transmembrane helix 2, and it's proposed to carry information across the bilayer from the receptor domain to the signal transduction domain. Now, the structure of the helix is warp-like, and what that means is it has a very simple hydrophobic core, indicated here in red, with flanking aromatic residues which lock the ends of the peptide into the interfacial region. What that means is that the hydrophobic region in the middle keeps it in the bilayer, whereas the flanking aromatic residues lock the position to prevent it kinetically from moving out of that orientation. It's said to be warp-like because this resembles an artificial peptide that's been used to understand membrane behavior called warp. And now, so because we know this is relatively simple, the behavior of the peptide in isolation is a sensible model for proposing how different modifications may, in general, alter such a system. So that is how the mutants change the behaviors.
I didn't understand that. Can you just say that last sentence again?
Yeah, so because we know that this helix biologically resembles a model system which we can understand in isolation, because it's quite simple, we hope that we'll be able to, we hope it's a reasonable approach to just treat this peptide in isolation, to understand how it moves in isolation from the rest of the protein, to understand how it will move in the context of the whole protein. So because it's simple, we'd expect its own behavior to be dictated by
So you're really going to study the warp thing?
the warp-like peptide, yes, that's right.
Okay, you're going to ignore the rest?
For now.
Okay.
We'll get on to the bigger stuff soon. So the key problem that we have in the field of bacterial chemotaxis is that there have been vast amounts of experimental data generated over the past 30 years, where people have mutated almost every residue in the transmembrane domain to every other possible residue, and then measured the output in terms of either how the cell is swimming, how the downstream effectors are altered, and how the whole system changes. However, the problem with all the mutagenesis work is that the experiments are difficult to interpret in terms of the biophysical event of signaling. So you can't measure the actual structural changes which are induced by the mutant. So you can't unambiguously link a particular mutation to a particular biophysical event to a particular biological observable. And so what we would hope to do by looking through all of these mutants is to be able to assess how the biophysics, how the mutants alter the biophysical properties of the peptide, so make it move up and down in the membrane, and then relate that back to the biological observables and say, this is unambiguously the model for signaling.
How do you know what mutants to study? Because there's presumably a zillion of them.
Yes.
Like 2 to the more proton-derived universe.
So it's an interesting question. So the systems we started on, and we're going to talk about in a second, are the systems with a trend. They make the easiest sets to look at. So for example, one particular set has a trend where half the mutants are active and half the mutants are inactive. Now, the advantage of that is that you can then compare all of the various metrics we're generating for all these peptides back to that trend and see where the trends correlate. What's rather harder to look at is where you've got data sets where they've got, for example, from a genetic screen, where the mutations can appear anywhere, and the active patterns in activity and inactivity have no clear trend and are harder to interpret.
You answered a more complicated question than what I was asking.
Okay.
Just thinking, you've got this thing and you're looking at mutants,
Yes.
variations of them. Do you generate the variations, or do they come from other people's experiments?
They've come from other people's experiments.
So you've just got a fixed library of 200 mutants that you want to study?
Exactly,
Oh,
yes.
okay.
Yep. Though ultimately we would hope to use these to predict mutants to improve the experimental protocols, and I can talk about that later if it's of interest. So from all the mutant data that has been in the literature, and as well as crystallographic data, there have been 2 major models of how signals are passed across the membrane into the cell. One suggests this helix of interest, TM2, slides by about 1.5 angstroms or 0.15 nanometers towards the cytoplasm on attractant binding. So it's a very small motion, and this small motion then causes a downstream set of effects which lead to kinase inactivation. Now, in contrast, this has been supported by a variety of different experimental methods, but in contrast to that, recently a model has been proposed where this same helix, rather than undergoing a sliding motion, undergoes a very small rotation of about 26 degrees in the membrane. And this small rotation then causes a clockwork-like set of rotations in the downstream physical machinery, the HAMP domain, the signal transduction domain, and then this causes the downstream effect. And so what we really like to do is to address this large body of mutants and say, do they favor one model or do they favor the other?
When you say model here, you mean something slightly different than
Yes.
what
So when I was talking model earlier, I was talking about a structural model. So that is the 3D positions the atoms in space. Now I'm talking about a model of the mechanism of signaling. So that is the proposal that the helix undergoes a sliding motion or a rotation motion. So it's a different kind of model, but it's related to the others.
What were these based on?
These are based on mutational data and crystallographic data. So we have for example, for certain structures, we have the 3-dimensional positions of all the atoms of most of one particular component, and they appear to have been moved by about 1.5 angstroms and in the ligand bound versus the not ligand bound. And so it's proposed, for example, from that crystal structure, that there was this shift. Furthermore, you can actually experimentally, you can label different parts of the protein, and you can see if you can observe that 1.5 angstrom shift. But because it's so small, it's typically hard to access on experimental scales. And it's very hard to get a decent signal compared to all the noise you get. So some of the measurements are not necessarily on the transmembrane domain, which is what they're proposed to affect. So there's a variety of different information, but it's all generally indirect. And also, there's the interpretation of the mutant data, which is obviously open for debate, or potentially open for debate. So what we start off by is looking at the aromatic residue mutants of the aspartate receptor TAR. And in particular, we've looked at a tryptophan tyrosine asymmetric scan, which produced a very distinct trend in behavior. So these are aromatic residues which anchor the peptide in the membrane. So they produce specific interactions with the interfacial region, that is the edge of the bilayer, and therefore they're proposed to either pull the helix up or down, depending on if they've been moved up or down in the sequence. We've also looked, experimentally, they also looked at removing some of these anchoring regions and replacing them with alanines. And in each case, I've run 100 nanosecond Martini simulations for each mutant that's available. And that's a standard procedure for this. To give you an overview of what this data looked like, what you have is the sequences of all of the transmembrane regions which were studied by the group. And what you can see is, in each of them, they move this WY motif up and down the sequence from the wild type, which is in the middle. And what they observed is that the kinase activity increased when you moved it towards the C terminus at the top, and decreased when you moved it towards the end terminus here at the bottom. This is the lab experiment with which we're comparing. So the interpretation of this result was that the helix is pulled up and down by the specific interactions of the tryptophan tyrosine with the interfacial region. And that was it. So the idea is that at this position, the helix is pulled towards the cytoplasm because the tryptophan tyrosine has been moved up in the membrane, so it's drawing the helix down. However, the problem with it being a helix is that as well as changing the position in the membrane, it also changes the rotation along the helix. So this also potentially could change the rotation and the tilt behavior of the helix as well. So that's the thing we explicitly looked for. When we looked at the bilayer position, however, what we observed was that the active mutants and the inactive mutants were both displaced relative to the wild type in the bilayer, with the inactive mutants being pushed towards the cytoplasm, active mutants being pushed towards the periplasm, in good agreement with the piston model, this concept of the helix just sliding up and down. They're both shifted by about 1 to 1.5 angstroms relative to one another, which is a good and an unexpected result.
This is the result of the
This is the MD results, yes, or one of the MD results. Furthermore, when we looked at the rotation and the tilt behavior, there was no correlation between the trends that were observed in the experiments and the tilt or rotation of the helices. So that is, we can only see a correlation between the change in the position and the biological activity. And so what we propose from this is good support for the piston model, where there's just a sliding of the helix up and down. Alongside this, we looked at the different set of mutants where this tryptophans the anchoring tryptophans were then removed from the sequence and replaced with something which doesn't have the same anchoring effect. One mutant where it was removed mimics the previous system where we mutate the same tryptophan, we remove that tryptophan and cause the downstream kinase to be inactivated. If we remove the tryptophan at the other side of the membrane, we only get a very weak inactivation, and then when we remove both of them, we get a mixture of the behaviors that are observed in both. When we look at this data set, what we see is actually there's no change in position here. What we do see is there's a very distinct change in the tilt of the helix in the membrane. And this tilt now correlates with the kinase activity. So the individual point mutants cause an increase in the tilt about 2 or 3 degrees, whereas the double mutant causes a tilt of roughly 5 degrees in total. So these are very small motions, but very consistent with what the experiments have told us so far. Again, looking at this data set, there was no change in the rotation behaviors observed. So again, there's no correlation between rotation of these helices and the experimental observable.
So the correlation is causation. Just because it's tilting, that doesn't mean that that's what's causing the difference. Yeah, it could be something else.
That's true. I think it's what exactly is going on in the membrane is something we can only really understand when we've got both this kind of data, which suggests that it might be a pushing type force, and the data from the complete set. Because then from what we'd hope is being able to relate the behavior on a very small scale to the behavior on a larger scale, that we'll be able to take those two results together and understand how they're causing the downstream signaling. So even if there are these small motions, we need to understand how these small motions then may lead to the downstream effects. Yes?
Aren't we missing here the information regarding, for each of these mutants, what happened in activation and in inactivation? Doesn't it need to, in addition to being tilted, be able to move inside the membrane?
At the moment, it's not known how. So, I think if I understand your question correctly, you're suggesting that the tilt is not enough. Actually, that there may be external forces applied from the whole structure.
There's a tilt.
Yes.
You're saying the tilt causes inactivation, but don't you need to have in addition that? Don't you have it to show in addition that on a specific mutant that is tilted in a certain way, there should be an additional movement when it is activated or inactivated?
So we're not able to experimentally determine that kind of observable. And I know people have tried to do this by specifically labeling it with techniques such as solid-state NMR. The key problem and the reason why this kind of approach is valuable is that you can't address this experimentally. You can't actually assess the position and orientation of the helix in the bilayer experimentally. You can only look at downstream experimental observables. So there is no direct way of getting this kind of insight. Now, I appreciate what you say. We need to both observe it observe it both in the larger system and in the smaller system. But the problem is the larger system is too large to get this kind of good statistical data. The results we get from the larger systems typically only come from a single MD simulation, and so not really statistically valid in the same way. They're still qualitatively useful, but they don't produce the same kind of information. So without wanting to go too much longer about the Sidekick side of things, we also looked at two other sets of information. One, principally now where they introduce basic residues, both into the interfacial region, which we were talking about previously, but also into the hydrophobic core of the membrane. And this has been done by site-directed mutagenesis for tar from Salmonella and the related protein trig, which protects glycine in a genetic screen. The interesting results are here that the results from Miller, the site-directed mutagenesis, indicated that there was a very distinct piston, swinging piston type motion. And this was the conclusion of the experimentalists. Now, in contrast, the genetic screen data didn't produce a clear interpretation, and it wasn't really interpretable. An interesting property, though, was that basic residue introduction into the hydrophobic core caused two distinct effects in adjacent positions. When we look at the Miller data in simulation, what we find is that they really reproduce what we've seen previously, a swinging piston type behavior, exhibiting both changes in tilt and position in agreement with what we would expect from the previous results. And this is just a quick demonstration of that with the graphics. When we look at the genetic screen data, this is rather more complicated, but because what we see here for the two neighboring positions, which we get different activities, we see the same behavior in terms of displacement. So this seems to contradict the results we've had previously. However, the one oddity about this result is that when we look at the rotation now, the kinase active states both have a much stronger preference for one particular direction, but also are rotated roughly 180 degrees compared to both the wild type and the kinase inactive mutant. So for this specific case, what we propose is happening is actually that the system may be broken by this extreme rotation which hasn't been observed previously. That the rotation here is much larger than any rotation which has been proposed based on the structural data that's been available. And actually, this is an unnatural event leading to the biological readouts. And these are circular histograms where the colors indicate the population of a particular rotation direction. So reds are very highly populated, blues are unpopulated. You can see there's much stronger preference for one particular direction for the three basic residue mutants relative to the wild type, but also that these active mutants are facing completely the opposite direction to both the wild type and the kinase inactive mutant. And this is the same data represented in a slightly more conventional format. And so what we're left with is a signaling state hypothesis where we say that the helix slides down in the membrane in response to a attractant binding event, increasing in tilt and changing in position, leading to downstream kinase inactivation. We can't rule out at this stage that this transition may require the helice's ability to rotate, particularly in light of the result from the Baumgartner data. And also it's worth saying that in the context of the complete system, we may not be able to separate the mechanistic difference between the changes in tilt and the position because of the fact there's a wider structure there which may change these behaviors. So that's all I'm going to talk about today with regards to the high throughputs. The next set of data I'm going to talk about is the dynamics of the complete structure. And the complete structure represents a real problem for molecular modeling. And that is because typically, where you've got large stretches of missing structure, they cause major problems for molecular modeling type approaches. And in particular, the transmembrane domain is structurally uncharacterized at high resolution. So this creates a sort of big gap in our knowledge, which normally would be a challenge. Furthermore, with regard to TSR, it is large, it is dimeric, so that is the individual covalently linked chains always exist as a dimer. Those dimers frequently exist as a trimer of dimers, so that's 3 times 2. These trimers of dimers then exist in a very large hexagonal array covering about 22,000 nanometers on the cell pole, and which communicate information throughout the array. And in addition to this, there are other key regions that are missing which connect the different domains, as well as the transmembrane region. So it really poses a unique problem for us.
So there are 2 problems. One is scale, there are a lot of atoms.
This,
The second is we haven't got a clue what some of the atoms are.
So I would characterize it slightly differently. We don't know where the scale is a problem in terms of getting the amount of resource and treating it appropriately, so running it long enough so we can understand if it works or it doesn't work. I think there are missing data regions, but I think the third problem would be there is also, we also know that there is a larger structure that we're not able to model. So for example, beyond a trimer of dimers is very complicated to model because people don't understand how the hexagonal array packs. So there isn't experimental data there, so we're unable to model that, and so we've got to be aware that we're explicitly missing out that aspect.
And what the molecular modeler does, you just say, here are all the atoms now, let them settle down.
We can put any atoms in any place. The problem is it's validating them after the event, because some simulations will allow you to discriminate between good contacts and bad contacts, but many simulations won't give you that kind of predictive power. So they might be able to tell you if things are falling apart. And the more complicated the system is, and the larger the system is, the less likely you are to be able to be confident that what you're observing is real. So what we do here is we try to exploit what we term as self-assembly. That is, we allow disordered regions of the structure to equilibrate in the hope that they will fold into roughly the right orientation. In particular, we do use this for the transmembrane region when it was modeled as attached to the receptor domain. So we use the receptor domain to initially see the positions of the transmembrane helices, and then we allow them to come together over a period of time. By running simulations of small pairs of domains and individual components, then we use homology modeling to stitch these individual components together and reinforce the original structural data onto the system. So we hopefully reproduce something realistic. Homology modeling is where we take similar structures that are available in the literature to the sequences we're interested in, and we use those similar structures to build a structure of the thing we're interested in, even though we don't have any direct structural information on the data that's available. So it's a way of taking data from experiments and...
It looks like something we've seen before.
Okay. By using this domain, taking the domain pairs and using the overlap, we've been able to generate two different models of the structure. One where we've imposed symmetry explicitly, which resembles the one on the left, which very closely resembles the pictures you will see in the literature of how the structure is proposed to look, but also an asymmetric one where symmetry has not been imposed, which seems to have a bending around the HAMP domain. And it's really not clear from the offset which of these would be experimentally favored, even though one is the picture that's used in the literature a lot of the time. So we ran simulations with both of them. The simulations we ran were the coarse-grained molecular dynamics I've talked about already. And in this case, we're using the molecular dynamics to both validate the structural models, but also to gain insights into the dynamics of the protein and relate these dynamics to the protein function. And in each case, we ran 4 replicates about 5 microseconds, so these relatively long simulations compared to the very small simulations before. What we observed is the structured domains are stable and largely maintain their folds. This is a good validation because the protein is not falling apart. What's more, we see a bending observed along the length of the protein, which generates an asymmetric receptor, regardless of whether we start with a symmetric receptor or an asymmetric receptor. So bending always occurs. This bending is at we've identified as being at the HAMP domain, or neighbors the HAMP domain, and could potentially be very large, up to 60 degrees, though the modal position is about a bend of 30 degrees. So really, we had to investigate this, and we had to validate this with experiment. This has not been something that had been proposed previously. When we went over and combed through the experimental data, what we found is actually there's lots of disparate data which seems to suggest there may be a bending event. The best data that's available comes from lipid nanodisk preparations. So these are little patches of membrane which are able to be cut out and in which you insert a single protein. What we observe when we look at the EM data is that you can see individual nanodiscs, which is the flat region here that I'm highlighting, and you can see an individual receptor dimer in this nanodisc, and if it was symmetric, you would expect it to be at 90 degrees to the bilayer. What we actually observe is there is a bending around HAMP. This is the clearest image from that particular still, but actually this has been observed throughout all the dimers that have been prepared in EM. So this is probably the best evidence that this is real. Alongside this, we know that the kink region correlates to a known area of processing sensitivity, so a region which we would expect to be more bendy. And furthermore, upcoming unpublished cryo-electron microscopy data seems to indicate on the whole array, seems to indicate there may be this kinking below HAMP, but that's rather more difficult to quantify having not been published. We can then go back and try to validate the structural models with other experimental data that's available, particularly the transmembrane region for which we didn't have high-resolution data. What we find with disulfide mapping experiments, our results are broadly consistent. So the orientation of the helices agree well, though the positions may be slightly different. And the orange regions here, which face one another, indicates regions of high efficiency cross-linking, which means these regions should face one another in the structure, and indeed they do. When we look at the HAMP domain, which has an unusual internal packing in its available high-resolution structure, what we see is that the key interface is maintained. So the interface that we know to be sensitive to mutagenesis is maintained throughout the length of the simulation. However, the packing has been arranged subtly to give what is known as a knobs into holes packing, a classical packing for coil-coil regions, as opposed to the experimentally observed knobs to knobs packing, which is relatively unusual. And this has been achieved by helix sliding of one of the second helix, AS2, towards the coiled region below it. And what we think is that this is actually giving rise to the observed asymmetry and bending in the structure. We can then try to further validate this with an alternative modeling approach, and this is elastic network modeling. I'm not going to go into depth on this, but this is another low-resolution technique for understanding long-term dynamics, and these really further support kinking around the HAMP domain, identifying the hinge between two rigid bodies in the GNM, indicated by the region without any, the green region between the red and the blue region, and the anisotropic network model, which seems to, the arrows here indicate the bending motion along the length of the protein, focused in on the HAMP. A Gaussian network model. It's a type of elastic network model which is one-dimensional. I can talk about that later. I'll be very happy to. It's specific. It's like normal mode analysis if you're familiar with that. Okay. So, however, the dimer is only one interesting structure, and there are other interesting structures too. So the trimer of dimers is known to activate the downstream kinase, and so is thought to be the minimum subunit required for signaling. However, the bending of TSR, the TSR structure we observed, makes a free self-assembly where we just have 3 of the proteins in a box and let them come together difficult, because the degree of bending requires we have a massive, massive box to do this. So what we did was we started from initial structural model based on the crystal contacts, so think experimental observables, and we restrain those crystal contacts. What we observe is a zipping along the length of the structure, which occurs very quickly to form a rod-like structure, which is retained after the restraints are removed. To show you what I mean by that, if we start off with this tent-like structure with the individual dimer rods coming away to form a rough triangle with a space in the middle, what we see after 500 nanoseconds is they very rapidly coalesce to form a single bundle and single rod. We can then compare this directly to experimental evidence, and in this particular case, we're lucky enough that there is something direct to compare with. Cryo-electron tomography results seem to indicate that our model, which shows a bundle stretching from the core region here all the way to the transmembrane and receptor domain here, agrees nicely with the experimentally observed APO, low-resolution APO structure, where you can see the bundle stretches from the tip of the core to the transmembrane region here. Furthermore, if you were to look at the ligand-bound structure, which we've not simulated, you can notice that there is a break between the HAMP domain and the coil domain, which would seem to indicate there may be a value, the bending we observe in the dimers may be significant for the signaling process itself. Just to go quickly over the dynamics of the trimer of dimers without restraints, what we find is that the structure and the contacts are stable even after the restraints have been removed, and we still show some bending around the HAMP domain. So how do we interpret these two different data sets and 2 different behaviors? Well, the structure is highly dynamic, so it's certainly in isolation, it's very free to bend and very free to move. And so what we propose is actually that this dynamic behavior is indicative of a general model whereby the HAMP hinge is tightened or loosened to create the bending and to cause downstream kinase inactivation. And so what we propose on the basis of this is other modifications that we know happen in the cell alter the wider structural stability to tighten or loosen this HAMP hinge. However, this is very valuable for individual dimer and trimer of dimers models. We want to move beyond isolated structures, though, because the cell is a much more complex environment than the model systems we've had here. The model membrane environment lacks several elements we know are required for function. So the membrane that exists in is curved, there are a much more complicated mixture of lipids, and there are very high concentrations of proteins. And so what we've been able to do is we've been able to take the models we've generated and we've built them into very large 17 nanometer vesicle systems which can be simulated over a supercomputer resource. So these are the 15 million particle simulations. This is a nice experimental analog because we know that receptor arrays localized to the cell pole that is a region of high curvature in the membrane. We know that one particular lipid, cardiolipin, pulls at the cell poles and regions of high curvature in large isolated membrane vesicles, so that's or spheroblasts. Furthermore, we know that receptor dimers are not thought to exist in isolation in vivo, but trimers of dimers can loosely associate. So what we'd expect is that dimers have an intrinsic tendency to aggregate, whereas trimers of dimers should have a lesser propensity to aggregate. So we start off with an empty lipid vesicle. We run a realistic mixture of the lipids of importance, cardiolipin, DPPE, and DPPG, mimicking the experimentally determined quantities. What we see is, with no proteins there, these lipids remain mixed over the period of a microsecond of simulation, so they don't naturally partition. We then can take that vesicle that we've generated, and we can put the protein models we've generated into that, so we can distribute them evenly over the surface. And then we restart the simulation and run for 2 microseconds. To give you an idea what these things look like, the outside of the surface, you can see here on the left, the dimers sort of spot over the surface, creating quite evenly. And in this, the model on the left, on the right here, the trimers of dimers are equally evenly distributed. And in each case, we've got about 30 dimers for each similar system. Now, on the surface of things, it doesn't look very busy. But because these things are so long, what we find is actually the core is quite densely packed. So there's at least 2 nanometers between every dimer in the system, but there's quite a lot of room for interaction between them over the course of the simulation. What we see is that the protein dimers self-associate very tightly to form extensive contacts which mimic those that we've observed in our trimer of dimers model. So this is a very good validation for the trimer of dimers model we talked about previously. Furthermore, the trimers of dimers associate, but they make less extensive contacts. So the structures with a lesser propensity to aggregate don't make as big interactions with their neighboring proteins. Furthermore, we can make conclusions about the specific lipids, and what we find is that cardiolipin closely associates with the protein structures in the membrane in a specific fashion. And just to show you what happens over the course of the simulation, going back to this half shell where we remove the front half of the lipids to reveal the proteins on the inside, what you can see over the course of the 2 microseconds is that extensive bending and interactions are observed in the dimers here, where we actually see them start coalescing and wrapping around one another. Whereas in contrast, the trimers of dimers do interact, but they have very much smaller interaction surfaces. So how do we interpret this? Well, this validates the rod-like model of the structure of the trimer of dimers in the sense that it's stable and the dimers self-associate to generate model contacts. But beyond this, the specific cardiolipin contacts also indicate that cardiolipin partitioning may not be induced by membrane curvature, which has been proposed previously, but actually be induced by the protein interactions themselves, and the proteins themselves may move to regions of high curvature in the membrane. So finally, to sum up, I've generated coarse-grained models of the CSR dimer and trimers of dimers, and I've modeled over 2 decades worth of transmembrane helix mutants. The complete systems are stable, they're largely consistent with experiment, and show kinking at the ham domain induced likely by a swinging piston observed in the motions of small helices, causing a bending, a tightening or loosening of the hamp hinge. Furthermore, the dynamics may be indicative of a model of chemoreceptor activation through subtle changes in this hamp packing, this hamp hinge, which change the kink properties and turn the signal from an on to an off state. How would you go beyond this? Well, I think the results here are all, it's fair to say the results here generated are all largely structural, and the models proposed, whilst we've got specific quantitative data from the high-throughput simulations, have been largely qualitative in some senses. Whilst we have the position of all the atoms in space, the interpretation we're putting upon them is a generic bending interpretation rather than a specific number. So one of the further problems to this is that the direct experimental validation is difficult to, for a large extent, impossible given the current available techniques. So a lot of the things we've been talking about may be separated from any validating experimental technique by at least one layer of interpretation because of the actual experimental measurables. So really, to go beyond what we've got here, as well as to expand the system, we're also to explicitly address the network effects and see if we can relate the observables we see in our simulations to wider models of the network, see if we can predict experimental observables rather than just structural observables. And I just need to acknowledge the various people who've worked with. This work was all done. This is work all done in Oxford with Mark Sansom and Judy Armitage. And I just want to thank all the people who helped out different stages along the way. And thank you all for listening. So at present, we're the only people who've been working on the high-throughput simulation of the transmembrane helices to the extent we've been working on. Now, there are other automated systems for running MD simulations. However, to my knowledge, Sidekick is the only example of something where people have tried to make an end-to-end pipeline. So it can all be, you can do all of the simulations and get all the analysis you want through the web server. You can get more data if you want to reanalyze the data, but we can actually, you can be completely shielded from the nitty-gritty of how the data is generated and to be honest, vast amounts of raw data that's generated. So I didn't mention it, but there's about 6 or 7 terabytes of data been generated over the past year by the Sidekick pipeline. Now, to make that manageable, you have to use the analytical metrics. With regard to MD of the complete structure, there are people working on individual components, but again, nobody has been looking to build the models of the complete structure because of long-standing issues with missing data regions. Because we've been able to overcome them using the coarse-grained folding approaches, we've been able to uniquely press on in this space. It's very likely, though, so the Sidekick data on the transmembrane helices has been published. Once we publish the work on the chemoreceptor, the complete model of the chemoreceptor is currently under review, and we'll make those models. When we make those models available, what I'd expect is there more people in the field trying to exploit the entire system.
I mean, since you're saying nobody else is doing sort of end-to-end result studies of this sort.
Not the particular method, not this particular methodology. So there are people working on this from a systems level. And so, for example, Dennis Bray and Karen Lipkoff have worked for a long period of time on models of how the trimers of dimers communicate information through the array. And equally, people have also modeled how the information is passed throughout the cell. And so that level of modeling has been going on, and that's been going on for a very extended period of time. But for the specific aspects I've been talking about, the structural modeling, there hasn't been as much available. Whilst there has been some data made available for particular components, they've either not done it through a high-throughput regime, or they've not looked at modeling a complete system in the same way. So the results we're able to generate this are very much unique to the approaches we've used.
So when you say you've got 7 terabytes of data,
Yes.
Is that something to shout about?
It is something to shout about, but it causes a lot of problems from a lot of different... It causes problems, but it also creates opportunities. So the problem is we have to store it, we have to curate it, and I've not talked about the technical details of how Sidekick has been implemented, but also we need a very big file system because the amount of data that can be generated requires... It is unusual to have that much data. But especially because of the way it's been generated in an automated fashion, one of the things we've not done yet, which would be very exciting, would be to try and mine this data. So having run individual simulations for a particular purpose, then to take all the data in aggregate and see if we can derive any new rules and new ideas from that data, or even pick out flaws in the model that are not obvious just from looking at individual data sets. That is something that would be very exciting to me. And that's why I think we have to keep the raw data, even if we only, for large numbers of particular problems, we only look at the metrics.